بسم الله الرحمن الرحيم. السلام عليكم. نواصل مع structure and function of hemoglobin. نلاحظ أن الهيموغلوبين يوجد بشكل خاص في اللي هي red blood cell، ووظيفته تكون اللي هي transport of oxygen from lungs to capillary of tissue. الهيموغلوبين يوجد بشكل عام in adult as hemoglobin A. It is composed of 4 polypeptide chain, 4 unit, 2 alpha chain or alpha unit, and 2 beta chain or beta unit, which held together by non-covalent interaction. Each chain or subunit has a stretch of alpha helical structure and hydrophobic heme binding pocket similar to that described for myoglobin. The tetrameric hemoglobin molecule is structurally and functionally more complex than myoglobin. For example, وظيفة الهيموغلوبين can transport hydrogen ion or hydronium ion and carbon dioxide from tissue to the lungs and can carry 4 molecules of oxygen خلال عملية التنفس from the lungs to the cell of the body. Structure of hemoglobin is quaternary structure. Can be envisioned as being composed of 2 identical dimers. The first dimer alpha beta, the second identical dimer alpha beta, and then 2 alpha beta identical dimer. In the same dimer, the 2 peptide chain in the same dimer are held tightly together by primarily hydrophobic interaction. It is strong interaction between alpha and beta, which get these 2 subunits held strongly together. The 2 dimer, alpha beta 1 and alpha beta 2, are held together by polar bond, which is nearly weaker than the first layer hydrophobic interaction. The weaker interaction between the dimers allows them to move with respect to one another. This movement results in the 2 dimers occupying different relative positions in deoxyhemoglobin as compared with oxyhemoglobin. The 2 form is first T form, second R form. T form, which is the deoxy form of hemoglobin, is called the T or tense form. The 2 alpha beta dimer interact through a network of ionic bond and hydrogen bond, it is a strong bonds. These bonds constrain the movement of the polypeptide chain. The T conformation is the low oxygen affinity form of hemoglobin. In the second form, it is R form or relaxed form. The binding of oxygen to hemoglobin causes by rupture of some of the polar bonds between the alpha beta dimer, decrease the number of polar bond, store some of the polar bonds, which is decrease the bond between alpha and beta dimer, this allowing movement. The R conformation is the high oxygen affinity form of hemoglobin. Binding of oxygen to myoglobin and hemoglobin. Myoglobin, which contain only 1 heme group, it can bind to only 1 oxygen molecule, while for hemoglobin, which is contain 4 heme group, it can bind 4 oxygen molecules. Degree of saturation of oxygen binding site on all myoglobin or hemoglobin molecules can vary from 0 up to 100%. Oxygen dissociation curve. This curve is a plot of a degree of saturation of oxygen on the y-axis versus the partial pressure of oxygen on the x-axis. In this curve we can find two important areas. المنطقة الأولى اللي هي at low pressure. At low pressure نلاحظ أن الهيموغلوبين can bond strongly with oxygen. Its affinity is high with myoglobin. بينما with hemoglobin بتكون أقل من ذلك. فلو أخذنا for example at 15% أو 15% of saturation for myoglobin, it is at low pressure, but for hemoglobin it need high pressure. So at low pressure, this pressure is in tissue. At this pressure, myoglobin can bend with oxygen, بينما hemoglobin release the oxygen which is transferred or carried. At high pressure, we find that the affinity, the saturation of oxygen at high pressure, it will be highly because the high pressure, high concentration of oxygen. So the saturation of oxygen for myoglobin, for the hemoglobin is high. But we know that hemoglobin can bend to 4 oxygen in contrast to myoglobin which can bend only oxygen. So العملية تكون كالتالي. في اللي هي lungs ضغط الأكسجين concentration of oxygen يكون عالي، وبالتالي يحمل الهيموغلوبين 4 جزيئات من الأكسجين transport this oxygen for the tissue. In the tissue the pressure is very low, so hemoglobin will release these oxygens and myoglobin will bend for these oxygens. The higher the oxygen affinity, the more tight the oxygen bend, the lower the P50 or pressure 15. Myoglobin in general plus oxygen will produce myoglobin oxygen molecule. The equilibrium in this reaction is shifted to the right or to the left as oxygen is added to or removed from the system. If we adding oxygen concentration increased and the reaction moved for the forward. But if the percent of oxygen, concentration of oxygen decrease, the reaction will be reversed toward myoglobin alone. From myoglobin bend released by hemoglobin at the low pressure of oxygen found in muscle. Myoglobin in turn release oxygen within the muscle cell in response to oxygen demand. إذا بعد ما يحمل الأكسجين أو الميوغلوبين الأكسجين من الهيموغلوبين عند الضغط المنخفض، نلاحظ أن الميوغلوبين ينقل هذا الأكسجين إلى الخلايا التي تطلب هذا الأكسجين خلال عملية اللي هي البناء الضوئي وخلال عملية التفاعلات الكيميائية التي تتم. هيموغلوبين أكسجين ديسوسيشن كيرف بشكل عام يمكن أن نلاحظ النقاط التالية على هذا الكيرف. The curve is sigmoidal in shape, indicating that the subunit cooperate in binding oxygen. The first, second, third, and the fourth subunit, it is cooperate in binding oxygen. Cooperative binding of oxygen by the fourth subunit of hemoglobin means that the binding of an oxygen molecule at one heme group increase the oxygen affinity of the remaining heme groups in the same hemoglobin tetramer. We will find that the first oxygen molecule increased affinity. The second oxygen molecule bent to the second unit increase the affinity more and more. The third will increase the affinity nearly up to 300% the speeding of binding of hemoglobin to the oxygen molecules. This effect is referred to as heme-heme interaction, as we will see in the coming slides. Allosteric effects. There is many effects that affecting the ability of hemoglobin to reversibly bind oxygen. The first factor is oxygen pressure through heme-heme interaction, and we will find that this effect is directly proportional, as increasing the pressure, increasing the interaction. The second is the pH of the environment, and it is reverse by increasing the pH. By increasing the pH, we will find that the hemoglobin affinity for oxygen decrease, the same as the pressure of carbon dioxide, and also as of 2,3-bis phosphoglycerate.